Hello everyone, my lecture will be devoted to the development of anti-cancer drugs based on nanoparticles and protein molecules. My name is Victoria Shipunova. I'm working at Laboratory of Molecular Immunology in Shemyakin Ovchinnikov Institute of Bioorganic Chemistry. You're welcome. Hello everyone, my name is Victoria Shipunova. I'm working at Laboratory of Molecular Immunology at Shemyakin Ovchinnikov Institute of Bioorganic Chemistry. And today I'm going to tell you something about the development of anti-cancer drugs based on nanoparticles and different protein molecules. And you are welcome to ask questions during the lecture in the YouTube channel during all the lecture. So in this slide you can see some types of nanoparticles which already entered the clinic. Some of them are used for diagnostics and some of them are used for therapeutic applications, for example for treatment of anemia or for MRI contrasting during diagnosis of some diseases. About 20 types of nanoparticles is already approved for applications in humans. And some of them are under development and in ongoing clinical trials. A lot of nanoparticles are used in clinical trials for the treatment of tumors, which enter the tumor due to enhanced permeability and retention effect. It is an effect caused by liquid vasculature of tumor. So tumor is a kind of organ in the organism, and tumor cells requires much more compounds than normal cells. And there are a lot of blood vessels in the tumor site, and the number of these blood vessels is much higher than in normal tissue. And when the nanoparticles enter to the bloodstream, and when it reaches the tumor site, it is retained inside the tumor. So this type of effect is called TPR effect, but one paper which is presented in this slide claims that EPR works in rodents but not in humans. What does it mean? Some types of nanoparticles which already come into application for treatment, for example, of breast cancer or ovarian cancer, for example, calyx or myoset. It is a liposomal form of doxorubicin, standard chemotherapeutic drug, works only based on this EPR effect. So it is a kind of passive transport to the tumor site and treatment of tumor cells due to the accumulation in the tumor area. And all fundamental studies are directed towards the development of such kind of nanoparticles that will enter the tumor and retain there and kill the tumor cells. But now there is an evidence that this effect fails in humans, and there is a need to develop some more intelligent, more smart biosystems that would penetrate into tumor and retain there, not only based on this well-known EPR effect, but based on other types of biological mechanisms. And so it is our lab, and it is my hand with magnetic nanoparticles, with a magnet in my hand, and We believe that this kind of nanoparticles is that type of the agent that will introduce, that will give rise to the development of new type of medicines for the diagnostics and for treatment of cancer. So the direction of Laboratory of Molecular Immunology, the main research focus is on the development of different targeted nanostructures. So we are trying to realize a magic bullet concept. Magic bullet, it is a real scientific term, which was introduced by the Paul Ehrlich, who won the Nobel Prize in physiology and medicine in 1908. He is an inventor of the first chemotherapeutic drug, Salvarsan. In his research, it was called compound 606. So it was really 600 compounds before that that he developed to introduce the first chemotherapeutic drug. And he was dreaming about such kind of ideal agent. It doesn't matter on the origin of this agent. This ideal agent should enter the tumor site and does not affect any other types of normal, non-transformed cells, do not have any side effects, do not affect liver, spleen, and other organs of the organism, in order to act as a smart agent only for tumor site without any side effects like cardiotoxicity, hepatotoxicity, and so on. And the concept of development of monoclonal antibodies, which was Nobel Prize once again, really helps to develop this concept of magic bullet. So now there are a lot of antibodies like trastuzumab and some others targeting the receptors on the cancer cells. So some cancer cells are characterized by overexpression or different expression of cell surface receptors. And these are cell surface receptors like HR1, HR2, and some others can be used as molecular targets on the cancer cell surface. And now some conjugates of monoclonal antibodies with toxins entering the tumor cells are already marketed by Roche and some other pharmaceutical companies. But sometimes the effectiveness of such kind of therapy is often non-sufficient for the disease treatment, and the therapy with monoclonal antibodies is often combined with standard chemotherapy and so on. And there is a big goal of all researchers in the modern nanobiotechnology and biomedicine to develop such kind of therapeutic and diagnostic agent, like picture illustrated in the slide here, possessing some kind of sensing module, for example, recognizing molecule or small substance that will bind the cancer cell receptor. Possessing some kind of payload, for example, chemotherapeutic drug, for example, doxorubicin, and so on. and some kind of engine that can act as a smart agent on demand where it is need, for example, only in the tumor site when it is necessary to bind the cell and to release some kind of therapeutic payload. And nanoparticles of different origin are the best platform. It is ideal tool for the design of such kind of diagnostic and therapeutic agents. And moreover, it is an ideal platform for the development of Theranostic agent. What is Theranostics? It is a combination of diagnostics and therapeutic properties on a single platform, on a single agent. It is one kind of agent that capable to make diagnostics and treatment where it is needed. Now there are a wide spectrum of organic and inorganic nanoparticles like magnetic particles, gold, silver, liposomes, and so on. In our laboratory, we are working with maybe 80% of existing nanoparticles, and it is not the finish. Why nanoparticles? Why not the conventional protein molecules? Why not antibodies? So nanoparticles, it is an object with a size from 100 nanometers to maybe 200 nanometers usually. And the high proportion of surface atoms on the surface of nanoparticles capable to introduce some new and original properties in this sample, which is not inherent in bulk samples. For example, superparamagnetism and magnetic nanoparticles, fluorescence and small nanodiamonds, and so on. So we have a lot of properties which are not accessible in bulk samples, in big samples, or in atoms and molecules. And there are a lot of other properties that can help to understand why different types of nanoparticles are much better than the conventional drugs. For example, widely used chemotherapeutic drug paclitaxel is poorly soluble in water, and it is administered into bloodstream in very toxic compound which may cause allergic reactions and so on. But incorporation of paclitaxel in liposomal formulation make it absolutely soluble inside this structure, and the problems of side effect is go away. My favorite nanoparticles historically, when I came in the lab of laboratory, in the laboratory of molecular immunology, was magnetic particles. These particles are actually tiny magnets, and in the absence of external magnetic field, they look like small balls without any magnetic properties. But when we turn on magnetic field, they look as they act as small magnets. This feature is due to the superparamagnetic properties of such kind of nanoparticles. and they can be synthesized by very wide spectrum of chemical and physical methods, but the most interesting for me and the most beautiful, as I think, the magnetosomes. It is a magnetic particles of biogenic origin. There are magnetotactic bacteria which are oriented in the magnetic field of the Earth when they are trying to find food and so on. So this type of magnetosomes can be like balls and other. So they can be of different shape, like here or here or here, and they are very they have very narrow size distribution, and they can be produced in bioreactors to get very uniform and batch-to-batch reproducible nanoparticles for different biological applications. Historically, the first application of magnetic particles was the studying of mechanical properties of living cells. For our cell is a kind of liquid structure with some organelles, nuclei, mitochondria, and so on. But it was found that the cell is very rigid structure. It was made using the big particles about 1 micron in size, using free magnetic poles and measuring the distance which the particle is going on inside the cells. Now magnetic particles are used for MRI contrasting, for magnetic separation, for different immunoassays, and maybe the most interesting application of these particles is to use them for the cancer hyperthermia. So magnetic particles can be heated by an external magnetic field, and cancer cells are much more sensitive to the heating than the normal cells. So when the particles is delivered to the tumor site, when the external magnetic field is turned on, the cancer cells in this site are heated, leading to cancer cell death and without any significant influence on the normal cells. So it is actively developing area in the targeted drug delivery and in magnetic nanobioengineering. And the main focus of our studies is the development of different structures capable to target cancer cells which overexpress HR2 receptor. HR2, it is human epidermal receptor 2. It is a member of EGFR receptor family, and it is overexpressed in about 30% of breast cancers and ovarian cancers. Moreover, it is often associated with poor prognosis for patient and with high metastatic potential of the tumor. So now monoclonal antibodies, trastuzumab, is marketed by Roche and our Russian company Bioorganic. Is used for the treatment of such kind of cancer, but it's still not sufficient for the full regression of the tumor. It is one work made in our lab. We developed magnetic particles capable of targeting HR2 overexpressing cancer cells using magnetic core for the diagnostics and therapeutic application depicted here. It is ferrum-free or for nanoparticles, and we modify them with targeting molecule. I will tell about this kind of molecules a little bit later. Terpene recognizing HR2 receptor, and we combine this diagnostic and therapeutic core with HR2 recognizing protein using Barnase-Barstar protein pair depicted here. Barnase is a bacterial ribonuclease, and Barstar it is natural inhibitor. These proteins are very, very small, 10 and 12 kilodaltons, so about 1–2 nanometers in size. And this particle is about 100 nanometers. So they can serve as a small adapter system capable to modify the nanoparticles with different protein and non-protein molecules. This small peptide was taken from the proteins from this bacteria, magnetotactic bacteria, which plays a key role in the formation of magnetosomes. So we take one short fragment from the protein from magnetotactic bacteria, combine it genetically, using genetic engineering methods, combine with one part of protein adapter systems, and using a kind of Lego approach and using this nanoparticle and this targeting molecule as Lego bricks, we just mix them to target cancer cells. And so it is electron microscopy of such nanoparticles, very beautiful particles of magnetite. And we use this fully assembled structure to target cancer cells. So white dots here, it is nanoparticles, so it is control cells. So you can see damaged cells with a lot of magnetic nanoparticles on these pictures, demonstrating how this approach is working in cancer cell targeting. So it was kind of rigid nanoparticles, nanoparticle synthesis for targeting cancer cells. Now we are working with many other types of nanomaterials, like polymer nanoparticles, and my favorite polymer is sopolymer of glycolic and lactic acid. See, these molecules are presented in our organism and making this biopolymer fully biocompatible and biodegradable. And now we can synthesize nanoparticles of this type of polymer, PLGA, very wide-known polymer for administration of different drugs. It is already FDA approved for about 20 compounds for diagnostics and diagnostic and therapeutic applications. So we used this polymer to load this with chemotherapeutic drug, and here you can see these nanoparticles, very nice particles of spherical shape. They're about 140 nanometers. As for me, I don't know any other research groups who can synthesize such small PLGA nanoparticles by using such very simple, like brute force methods, just using syndication of the solution, and you get your particles. Next, we modify this particles loaded with chemotherapeutic drug and one more label, fluorescent dye, just to visualize these particles in cells or in living organism. We modified these particles with targeting modalities capable to recognize receptors on cancer cell surface, namely HR2 receptor. Usually, scientists use full-size antibodies to direct nanoparticles to cancer cells, but antibodies, e.g., very big molecules possessing a lot of side effects like immunomodulation in vivo and so on. And its production requires mammalian cells because of post-translational modifications. So now biomedicine and nanobioengineering move to the next class of targeting molecules like scaffold polypeptides. They're very small. Here you can see two types of these peptides, like terpenes and affibody. They are fully artificial proteins which can be produced in bacterial systems, for example, in E. coli. And they are about 2 or 3 nanometers in size, and they can be used for different genetic engineering manipulations because of the availability of N and C terms in the structure of both proteins. And it is quite easy to get different terpenes and different antibodies to different molecular targets by different display methods technologies. And we use in this work affibody HR2-342 directed toward subdomains 3 and 4 of HR2 receptor. And we modified these round-shaped nanoparticles with affibody and get stable nanoparticles. And you can compare here the label and efficiency of such targeted nanostructures for HR2 overexpressing cells. So green contour here is the labeling of cells with molecular affibody with small proteins, 8 kilodaltons. And red dots here, it is targeted nanoparticles loaded with chemotherapeutic agent doxorubicin and fluorescent dye capable to visualize these cells in vitro and in vivo. Next, we use these particles for in vivo imaging. And red fluorescence here, it is fluorescence of nanoparticles which were intravenously injected into the bloodstream of immunodeficient mouse with tumor overexpressing HR2 receptor. Here in the red circle, you can see the fluorescence of targeted nanoparticles, and here is the absence of fluorescence of non-targeted nanoparticles. So the specificity of nanoparticles in vivo of such targeted nanoparticles is compared to the in vitro efficiency. As well so these particles possess kind of cytotoxicity caused by loaded doxorubicin chemotherapeutic drug, but you can see here that we did not achieve the full cell death. So you can see here about 30% of living cells. And to improve the cytotoxic abilities of the developed polymer targeted nanostructures, we add one more scaffold protein, namely derpin, which recognizes as a domain of HR2 receptor, namely subdomain 1. So Affibody binds to the domain 3 and the domain 4, and the second scaffold proteins binds to the subdomain 1. So they did not possess steric hindrance during the target recognition. And this targeting scaffold protein, derpin, was fused with low immunogenic fragment of Pseudomonas exotoxin A. So once again, you can see here the labeling of cells with overexpression of HR2 receptor with such kind of molecule. And using these two types of compounds targeting only one cancer receptor, HR2, we observed a strong synergy of these two compounds leading to cancer cell death in vivo, moreover to full reduction of metastasis in mice. The tumor was inoculated into the right flank of mouse, and it has metastasis on the left side. Using this combination therapy, we do not observe any metastasis on the opposite side, so it is a very good treatment strategy when we can take a biopsy of a tumor and found that only one receptor is presented on cancer cell surface, and there is no any other molecular targets like HR or EpCAM molecule or so on. So we have only one target to only one receptor, only one target on the cell surface, and we can improve the therapeutic efficiency targeting only one receptor. It is quite a new research result, which was recently published in ACS Nano journal. And one more paper based on polymer nanoparticles are now in preparation. I like this work very much. So it is an kind of incremental work of the described work here. We developed fully Theranostic nanoparticles. So we have polymer nanoparticles loaded with fluorescent dye and chemotherapeutic drug, and when these particles bind to the cancer cell receptor on the surface of the cell and is internalized inside cells, only inside cell this particle becomes fluorescent. So we can observe fluorescence only inside target cells. So we can use the light with the red light to make diagnostics of tumor, and we can use the light with other wavelength to treat the tumor. So when we find our nanoparticles which are fluorescent inside the organism, so we can see using non-invasive diagnostics methodology that here we can tumor, here we observe metastasis and so on. And when we inject these particles into the bloodstream and they reach the tumor site and metastasis, we can turn on the second light with green light, which make this particle to product reactive oxygen species, which is toxic for cancer cells, and cancer cell is damaged, thus leading to full tumor regression. So here you can see the specificity of such kind of nanoparticles. In green circle, the tumor of one type with overexpression of one receptor, namely HR2. In red circle, the tumor of the second type with no expression of this receptor. The tumor in the green side have metastasis on the opposite side, as I described previously. So this kind of fluorescent luminescent tumor have metastasis here and here. And when we inject our nanoparticles with red fluorescence inside the organism, these particles are accumulated only inside the tumor with overexpression HR2 receptor, and not in the control tumor, thus confirming the specificity in vivo of such particles, which very big achievement of this work. And using the biodistribution picture of fluorescence of these particles, we can see where we have tumor, where we have metastasis, and when and where we should turn on the green light, which will excite the production of reactive oxygen species of toxic compound incorporated inside the structure of nanoparticles, and to realize their cell targeting, cell killing abilities. And using the irradiation with green light, we achieved full tumor regression in mouse treated with nanoparticles and irradiation. This mouse were treated with nanoparticles, but no with irradiation. And you cannot see any difference between the control groups and groups treated with nanoparticles, thus confirming that nanoparticles are non-toxic without any external irradiation. So we can make particles toxic when we want and where we want. So the two types of nanoparticles were described here. One of them are rigid and magnetic particles, one of them soft polymer nanoparticles. One of the main direction of the research of our group in our lab now, it is the development of different anti-cancer agents based on plasmonic nanoparticles. Here, 3 tubes with silver nanoparticles. We know that silver is something bright and white, but here you can see tube with green nanoparticles and with yellow particles, and we can easily synthesize now round-shaped nanoparticles or nanorods or even nanowires based on silver. And using a very non-standard method of nanoparticle synthesis, namely green synthesis, using very beautiful plant lavandula, we synthesized nanoparticles possessing diagnostic and therapeutic properties. I should mention here that usually plasmonic particles like silver particles and gold particles are synthesized using very toxic reducing agent like sodium borohydride or any CTAB or any others which are very toxic and should be removed from the sample by repeated rounds of centrifugation or any other types of purification. Using the water extract of such beautiful plant, we just mix this the quite non-toxic extract with silver salt. We get very nice particles without any additional purification steps, and modify them with targeting scaffold proteins, and achieved full tumor remission using the nanoparticles modified with anti-HER2 molecule and irradiation with external light source. When these particles are irradiated with external light, they are heated with very good heating capacity, and causing the heating of tumor sites, thus leading to cancer cell death. So you can see the toxicity for tumor cells of such targeted particles without irradiation. Here, significant difference between treated mice and non-treated mice. But the full remission was achieved only with irradiation with external blue light. Of course, some people may say that we use blue light, which is not in tissue transparency window. It is fully true, but this method can be used for the treatment of such diseases as melanoma or other skin diseases. But now we are developing different types of methods how to irradiate silver nanoparticles inside the organism without the external light source. And I will tell about this in maybe future lectures in talking about our future works. I tell you some about some studies directed towards the development of different nanoparticles, to the development of their formulation, and about the improvement of their cancer cell targeting abilities. But when the nanoparticles came inside the organism, our defense mechanisms of mononuclear phagocytic systems with macrophages try to eliminate all the objects which are injected into the organism and which are not familiar to the organism. And we are now developing different methods how to make the nanoparticles and any other big structures like kind of cells in the organism, how to prolong the circulation, to give the time to nanoparticles to reach the tumor area. One of the methods is based on the blockade of mononuclear phagocyte systems with different type of solid nanoparticles. So we just feed macrophages with huge dose of non-toxic nanoparticles. Macrophages are full and they cannot eat other types of nanoparticles. So we can inject inside the organism one type of non-toxic nanoparticles, and macrophages are out of the game, and the second particles which are toxic, targeting, and so on can reach the tumor site, and such kind of method can prolong their circulations by 2 or 4 times, which we can measure this parameter we can measure by our original method of magnetic particle quantifications, which was developed in Institute of General Physics by Petr Nikitin. And the second method, how to prolong the circulation of nanoparticles inside the bloodstream, is a very beautiful, very nice method called hitchhiking. It is real hitchhiking on the red blood cells, erythrocytes. We incubate erythrocytes with different type of nanoparticles. Some of them are sticky, and they are attached to the surface of erythrocytes. We inject, the erythrocytes were taken from the mouse, incubated with nanoparticles, and injected into the bloodstream of the same mouse. And these particles on the surface of erythrocytes are hitchhiked on the surface of its own, of mouse own cells, and the circulation is prolonged by many times. And such kind of delivery of nanoparticles on the surface of cells like erythrocytes can help to improve not only circulation of nanoparticles inside the bloodstream, but for treatment of different cancer diseases, for example, melanoma. In this paper, melanoma treatment were demonstrated with magnetic particles loaded with doxorubicin, and you can observe red dots, black dots here on the lungs of mice in a non-treated group, and the absence of these black dots, melanoma dots, in the treated group. These mice were injected with nanoparticles on the surface of red blood cells, and this helps to improve the treatment capabilities of such particles by prolongation of their circulation in the bloodstream. And the last research which I will be talking about today is the third method of prolongation of circulation of nanoparticles inside the bloodstream and enhancement of their performance in vivo, is the blockade of mononuclear phagocyte systems with self-blood cells. So the main idea to use anti-RBC antibody which binds to red blood cells and cause the damage of these cells, cause erythrophagocytosis, and the uptake of these erythrocytes with macrophages. And once again, macrophages are out of the game. They cannot internalize other type of nanoparticles or different other objects, and we can achieve the significant prolongation of any type of absolutely any type of nanoparticles which are injected into the bloodstream of the animal. So you can compare here the blue curve. It is a circulation of nanoparticles without the, this kind of cytoblockade, and the red curve, the circulation of nanoparticles with cytoblockade, with this kind of blockade of mononuclear phagocyte systems with RBC cells. It is a magnetic signal obtained in the tail vein of mice, which is injected inside the detection coil of MPQ device, like depicted here. So it is mice, and this tail is inside the detection area. So mouse is live, happy, and so on, without any significant influence on her happiness. This kind of cytoblockade allows us to increase performance of nanoagents in the organism and make them capable to target cells in vivo like they do in vitro. It is fantastic. It is very beautiful results, because when we open some papers in Google Scholar and see excellent results on cancer cell targeting in vitro, we can observe only a slight difference in vivo targeting. For example, specificity of nanoparticles in vitro can be more than 300 times, and in vitro 1.2 times between targeting and non-targeting structures. And we show that using such kind of method to prolong circulation of nanoparticles in vivo, we can achieve the same efficiency of targeting abilities of nanoparticles as they are in vitro. For example, we target CD4 positive cells in the blood of immunocompetent mice, normal mice, which are presented in every laboratory. And when we do not use any kind of blockade in vivo, we do not observe any targeting at all. It is blue histogram, flow cytometry histogram, cells labeled with anti-CD4 nanoparticles. The green histograms show how these particles are efficient in vitro in tubes in culture of cells. And when we use MPS cytoblockade, we achieve the same performance of magnetic particles for labeling CD4 positive cells in vivo like they are in vitro. So we can, using such a method of prolongation of circulation of nanoparticles in the bloodstream, we can use all fundamental research studies achieved previously by other groups for different nanoparticles, supramolecular structures, and so on, which were non-effective in vivo, but which were effective in vitro. Because there is a big problem in targeted drug delivery. It is a delivery inside the organism, into the bloodstream, the uptake with macrophages of different nanoagents. So this problem is now solved, and we can use this method to even prolong circulation of already approved nanomedicines for human use, because this RBC antibody is already one type of anti-RBC antibody is already used in humans for the rhesus conflicts in pregnant women for the treatment of these women. And as well, some of already approved nanomedicines for cancer treatment is already used. So it is our big dream just to combine these medications in order to increase the performance of nano agents for treatment breast cancers in human. And the last example, the treatment of the accumulation of nanoparticles in different models of cancer, of tumors in immunocompetent and immunodeficient mice. So you can compare the red and blue histograms on this slide, and in all cases the difference is very significant. So that's all. Here are my contacts. Thank you for your attention. I have some questions from the chat. The most famous drug that works like a magic bullet. Can you give an example? Of course, I can give an example. Trastuzumab is a well-known antibody which is already approved for human use, and it is directed toward HR2. What's going on? For HR2 receptor. It is depicted on the slide and is now produced by our Russian company Bioorganic, and I can use it in my research because it is very cheap and of very good quality. And one more example is trastuzumab emtansine, which is about maybe 10 times more expensive than the trastuzumab, and it is a real magic bullet. So it is a combination of targeting modalities and toxic modalities. Emtansine is a kind of toxin which penetrates into the cell only after binding with HR2 receptor on the cancer cell surface. Are magnetic particles used to treat human cancer, or is it the research stage now? Magnetic particles are used for diagnostics applications, and some clinical trials are going now. There was one clinic, as I remember correctly, in Germany that treat patients with magnetic particles using hyperthermia, but it is very dangerous method, and in Russia it is not allowed by our organizations. But now I don't know about the magnetic particles that already finished the applications, the clinical trials for application for treatment of cancer. They are used for treatment of anemia and for diagnostics. What's your secret to grow in small biopolymers? Is it necessary equipment or technology or magic? It is magic. Actually, I started my lecture from the compound Salvarsan, compound 606. I made 230 syntheses to get the smallest particles in my research. I think just kind of magic maybe, but this magic is reproducible. And it is maybe it's just a good equipment or good equipment, good chemicals, a lot of attempts to get such kind of particles. But what I want to add is that we now can load these particles by any type of small compound, of any type of fluorescent dye, of chemotherapeutic compound, and it doesn't matter whether it's soluble or not. So this kind of synthesis method is absolutely universal, and if you have any ideas on collaborations, you're welcome. Thank you very much.